Hi everyone, welcome back to the channel. In today's video, we're going to talk about bioprinting, a particularly innovative field within regenerative medicine. Now, to kickstart things off, here's something that you might have seen circulating on the internet recently. This is a 3D bioprinter in action, simulating actual bioprinting with hair gel, creating intricate tissue structures layer by layer. So, what exactly is bioprinting? Now, simply put, bioprinting is an advanced adaptation of traditional 3D printing techniques utilizing live biological materials. Now, the process involves the precise deposition of bioinks, complex formulations containing living cells and biocompatible matrices in order to construct functional tissue and organ structures. Now, bioprinting requires three components. These are the cells, the hydrogel, and the bioprinter. The cells in the hydrogel add up to compose what we know as bioink. Bioprinting is divided into three phases. These are the pre-processing phases, the processing, and post-processing. This we will look at later on in another video. In this video, we will be looking at what bioinks are. Now, bioinks are biological materials used in the manufacture of engineering live tissues. Now, the term bioink does not only refer to the cells used in manufacturing, but also the carrier molecules that provide support to the growing cells. Now, common carrier molecules included alongside the cells in bioink are biopolymer gels that act as 3D molecular scaffolds in order to promote cell proliferation. Now, for those of you who may not exactly be familiar with what scaffolds refer to a 3D dimensional structure that provides a supportive framework for cells to grow, organize, and form cell tissues. Now, scaffolds are essential in tissue engineering as they mimic the natural extracellular matrix, also known as the ECM, guiding cell behavior such as adhesion, migration, proliferation, and differentiation. Now, to kind of visualize all of this into a diagram, the components of bioink, they can largely be divided into cells, hydrogels, and growth factors. Now, the cells used can either be stem cells, such as pluripotent stem cells or mesenchymal stem cells, or differentiated cells, depending on the tissue printed. Pluripotent stem cells are a particularly potent type of stem cell that only exists in early embryonic development. Here, the term potent refers to cell potency, which is essentially a cell's ability to differentiate into other cell types. The more cell types a cell can differentiate into, the greater its potency. Pluripotent stem cells are very potent so as to have the ability to form all three of the basic body layers, including the ectoderm, the endoderm, and the mesoderm, and even germ cells. Now, there are two types of pluripotent stem cells. These are embryonic stem cells, also known as the ESCs, derived from the inner cell mass of the blastocytes, and induced pluripotent stem cells, which are also called iPSC, which are adult somatic cells that have been genetically reprogrammed in order to return to the pluripotent state. Mesenchymal stem cells, also known as the MSC, are adult multipotent cells that can differentiate into a variety of cell types such as osteoblasts, chondrocytes, or even adipocytes. Now, the hydrogels in the bioink serve as a scaffold for encapsulated cells. These hydrogels are typically biocompatible and provide the physical structure needed for cell attachment, growth, and function. Common hydrogels can be divided into natural and synthetic polymers, where materials such as perhaps gelatin or collagen, fibrin, or chitosan that closely resemble the natural extracellular matrix, the ECM, compose the natural polymers, and materials tailored for specific mechanical degradation properties, such as the polyethylene glycol or the polyvinyl alcohol, compose the synthetic polymers. Now, the growth factors in bioink aim to essentially promote specific cellular behaviors such as proliferation, differentiation, and tissue maturation. Now, these growth factors largely include the VEGF, the vascular endothelial growth factor, which promotes blood cell formation, the TGF-beta, transforming growth factor beta, which encourages cartilage and bone formation. Now, in our next video, we will be diving into the bioprinting processes, breaking down how these bioinks are used and to create functional tissues layer by layer, and the technologies that make all of this possible. We will also try to look at the current applications of bioprinting and its future potential in medicine. So be sure to subscribe and hit that notification bell so that you don't miss out the next part of our series on bioprinting.